Hi friends, today I wanted to discuss about the topic of locomotion and movement. This is a chapter that is quite interesting where we are going to study about the types of joints. We are also going to study about the different types of movement, the actin and myosin involvement in the muscles. All these are the topics that are involved here. Movement is one of the significant features of the living beings. So animals and plants exhibit a wide range of movements. The streaming of protoplasm in the unicellular organisms like amoeba is a simple form of movement which we also call it as cyclosis. The circular movement of protoplasm. The movement of cilia, flagella and tentacles are also seen in many of the organisms. So human beings can move from limbs, they can also move their jaws, eyelids, tongue, etc. Some of the movement results in change of place or location for the organism. Such voluntary movements are called as locomotion. Walking, running, climbing, flying, swimming, they are all some forms of locomotory movements. In plants you can see growth movements or movement towards the source of stimulus, like growing towards the source of water, growing towards the source of light, or growing towards the gravitational force or away from the gravitational force. They are all growth movements that you can notice in case of plants. The types of movements that we can notice are amoeboid, ciliary and muscular movements. In amoeboid movements is noticed in macrophages and leukocytes in blood. They exhibit amoeboid movement wherein there is a formation of pseudopodia by the streaming of protoplasm similar to that of amoeba. Ciliary movement is noticed in case of most of the internal tubular organs which are lined by ciliated epithelium. The passage of ova through the female reproductive tract or genital tract is also facilitated by the ciliary movement. The movement of our limbs, jaws, tongue, etc., they require muscular coordination and movement. So we call this type of movement as muscular movement. So muscle it is also called as sarcomere. It is a specialized tissue and it is of mesodermal origin. Even in the animal tissues you might have studied about this. About 40 to 50% of the body weight of a human adult is due to muscles. They have special properties. Muscles have special properties like excitability, contractility, extensibility and elasticity. Based on their location, muscles are of three types. They are skeletal muscles, visceral muscles and cardiac muscles. If you look into skeletal muscles, which are also called as voluntary muscles, you can remember the biceps. They are also called as striped or striated muscles. Why they are called as striped or striated muscles are because they have light and dark bands or striations. They are called as voluntary muscles as they move as per our will. They are also called as skeletal muscles as they are attached to all the bones and help in the movement. The structure of skeletal muscles or striated muscles or voluntary muscles is they are long cylinder shaped unbranched cells, multinucleated, that is they are having many nuclei, and the fibers are arranged in form of bundles and they exhibit dark and light bands. The location of the skeletal or striated muscles or voluntary muscles are limbs, that is forearms and hind limbs, that is arms and legs, tongue, body, face and neck. They show the striped or striated muscles. So the muscles of striated muscles work by contract quickly but can't remain contracted for long, so they easily get fatigued or tired. That's the reason the more you work out on your biceps, you get exhausted, and at a particular point of saturation point you can't lift the weights much. So just remember about these are due to the fatigue that the muscles undergo. Now the unstriped or non-striated or involuntary muscle fibers, they are called as unstriped or non-striated because they do not exhibit light and dark bands. They are called as involuntary as they are not under our control, and they do not exhibit voluntary movement. The structure of the smooth muscles or unstriped or non-striated muscles are spindle-shaped cells. They are uninucleated and the fibers are arranged in form of sheets. Dark and light bands are absent. They are usually located in the iris, ureter, bronchi of lungs, alimentary canal wall, blood vessels. They are all made up of the smooth or non-striated or involuntary muscles. The working is they contract slowly but can remain contracted for long. They do not get fatigued or tired. The cardiac muscle fibers, they are structured in between striated and non-striated muscle fibers, and they are involuntary. These muscles exhibit rhythmic contraction and relaxation throughout life, and they pump blood. The structure of cardiac muscle fibers are they are short cylinder-shaped branch cells. They are uninucleated. The fibers are arranged in the form of network, and they exhibit faint dark and light bands. They do not undergo fatigue and they are not under our control. The location of cardiac muscles are, as the name indicates, they are located on the walls of the heart. Working, they contract quickly and rhythmically. Therefore, they do not get fatigued or tired. That is the nature of cardiac muscle fibers, which are also involuntary muscles, and they also exhibit striations. The mechanism of muscle contraction. How do the muscle contract? If you look, each and every organized skeletal muscle in our body, it is made up of numerous muscle bundles. They are called as fascicles, and they are all held together. These muscle bundles are all held together by collagenous connective tissue layer, which is called as fascia. The muscle bundle contains a number of muscle fibers. So the muscle fiber is lined by a plasma membrane, which we call it as sarcolemma, and the cytoplasm of this muscle fiber is called as sarcoplasm. So the muscle fiber is a syncytium, as the sarcoplasm contains many nuclei. The endoplasmic reticulum of the sarcoplasmic reticulum of the muscle fibers is the storehouse of calcium ions. One of the characteristic features of the muscle fiber is the presence of large number of parallelly arranged filaments in the sarcoplasm, and they are called as myofilaments or myofibrils. Each myofibril has alternate dark and light bands on it. A detailed study of the myofibril has established that the striated appearance is due to the distribution of two important proteins, the actin and myosin. So you can look into the diagrammatic cross-sectional view of a muscle, which shows muscle bundles and muscle fibers. So you can notice the muscle bundles, they are all arranged together as fascicles. The muscle fibers or muscle cell, they have an outer membrane which we call it as sarcolemma, and the cytoplasm we call it as sarcoplasm. And you can notice the muscle fibers are all intricately connected with the blood capillaries. The light band it contains actin and is called I band or isotropic band. The dark band is called as A or anisotropic band, and they contain muscle fibers. So the muscle fiber is lined by the plasma membrane called sarcolemma, which encloses the sarcoplasm. Muscle fiber is a syncytium, as already discussed, as the sarcoplasm contains numerous nuclei. The endoplasmic reticulum is the sarcoplasmic reticulum of the muscle fibers, is the storehouse of calcium ions. One of the characteristic features, dark band and light band, we have again discussed about both the proteins. They are arranged as rod-like structures parallel to each other and also to the longitudinal axis of the myofibrils. So you can notice here the anatomy of muscle fiber in the A diagram showing a sarcomere. The B diagram is a sarcomere. So you can notice the Z line, and there is a dark band and light band. So the A band, that is anisotropic band, and I band, that is the isotropic band. So the anisotropic band is darker in color and the I band is lighter in color. So now the structure of contractile proteins. Each actin filament is made up of 2F or filamentous, where F stands for filamentous actins, which are bound to each other. They are intertwined with each other. Each F actin is a polymer of monomeric G actins. So the two filaments of another protein, tropomyosin, also run close to the F actins throughout its length. A complex protein, troponin, is distributed at regular intervals on the tropomyosin. In the resting state, a subunit of troponin masks the active binding sites for myosin or the actin filaments. Each myosin filament is also a polymerized protein. Many monomeric proteins called meromyosins constitute one thick filament. Each meromyosin has two important parts: a globular head with a short arm and a tail, the former being called the heavy meromyosin and the latter the light meromyosin, that is LMN, and heavy meromyosin is called as HMN, light meromyosin is called as the LMN. Now you can see an actin filament, thin filament A, and B is myosin monomer or meromyosin. So in the actin filament, which is a thin filament, you can see the two troponins are there, the F actins are interwound, and you can also see the tropomyosin. So you can notice here and there the troponins are also distributed as per the description of the structure. So the myosin monomer, or it is also called meromyosin, they have a head region and a tail region. So the head region has the actin binding sites and ATP binding sites, and it is almost looks like the snake hook. That part is called as the cross arm. So this structure is very important to understand further concepts. So now the stages in cross bridge formation, where you can see the first diagram, there is the actin filament and there is ADP and P, you can notice. So in the next diagram part of it, during the cross bridge formation, the myosin head comes in contact with the actin filament and there is a formation of cross bridge, and the sliding or rotation of the myosin head happens, and there is a release of phosphorus, inorganic phosphate, and ADP, and this you can notice, and as well as the straightening of the actin filament. The next is breaking up of the cross bridge. You can notice where the myosin filament becomes free, and again there is a formation of ATP, which again enters into the cycle. This stages in cross bridge formation, rotation of head, and breaking of cross bridge. So there is formation, rotation of head, and breaking of cross bridge is a cyclic process. Now in this first diagram that you can see here the myosin actin interactions underlying muscle fiber contractions. There is a thick filament and thin filaments are there. So the first step is the myosin head is bound to ATP and is in its lower energy configuration. The thin filament is there. So the myosin head has low energy configuration and they are found from the thick filament. The second step is the myosin head hydrolyzes ATP to ADP and inorganic phosphate, and it is in a high energy configuration. The myosin, the ADP and B are there, myosin binding sites are there, myosin head with high energy configuration is noticed. The third step, the myosin head binds to actin, forming a cross bridge that you can notice, and in the Next step is the ADP and inorganic phosphate is released and myosin returns to its low energy configuration, sliding the thin filament. The thin filament moves towards center of sarcomere. So then the binding of new molecule of ATP releases the myosin head from actin and a new cycle begins. That is how this cyclic process of myosin actin interactions takes place, which was in the previous diagram told in a very simplified format. Now the sliding filament theory of muscle contraction, that is movement of the thin filaments and the relative size of the I band and head zones at various stages of muscle contraction can be noticed. In a relaxed state, if you look into the head zone is wider, the I band is also wider in nature and the A band is there. So the Z line is there in between. Whenever the muscles contract, you can notice there is a reduction in the size of the head zone and also the I zone or I band, there is a reduction. The maximum contraction of muscles, there is no head zone and I zone, so it is almost completely overlapped. So this is the sliding filament theory of muscle contraction, wherein there is a movement of thin filaments and the relative size of the I band and the head zones vary in relaxed, contracting and maximally contracted stages. Now we'll proceed for the next aspect, that is the skeletal system. The skeletal system consists of a framework of bones and a few cartilages. They are grouped into two principal divisions, the axial skeleton and the appendicular skeleton. If you look into the axial skeleton, it consists of skull, which has 28 bones, cranium, which has 8 bones, facial, which is having 14 bones, ear ossicle 6 bones, vertebral column 27, hyoid bone is 1, vertebrae in adult is 26, thorax 25, sternum is 1, ribs are 24. So this is the axial appendicular skeleton. The appendicular skeleton, axial skeleton. Appendicular skeleton consists of the upper region, that is the pectoral girdle, forearm. Pectoral girdle is 4, arm and hands, 60 bones are there. So in the lower region, pelvic girdle has 2 bones, legs and feet they have 60 bones. So the total bones that you can notice in the skeletal system is 206. And the skeletal system also should be having joints. They are essential for all types of movements involving the bony parts of the body. The fibrous joints, the joints are of different types, fibrous joints, cartilaginous joints. The fibrous joints, they do not allow any movement. This type of joint is shown by the flat skull bones which fuse end to end with the help of dense fibrous connective tissues in the form of sutures to form the cranium. In cartilaginous joints, the bones involved are joined together with the help of cartilages. The joint between the adjacent vertebrae in the vertebral column is of this pattern, and this cartilaginous joint, it permits the limited movements. The synovial joints are characterized by the presence of fluid-filled synovial cavity that is present between the articulating surfaces of two bones. This sort of fluid arrangement shows considerable movement. These joints help in locomotion and many other movements. The ball and socket joint, that is between humerus and pectoral girdle, hinge joint, that is knee joint, pivot joint between atlas and axis, gliding joint between the carpals, and saddle joint between carpal and metacarpal of thumb. They are all examples of synovial joints. So now the last part of today's discussion on locomotion and movement is the disorders of muscular and skeletal system. So there is a disorder called as myasthenia gravis. It is an autoimmune disorder affecting the neuromuscular junction, which leads to fatigue, weakening and paralysis of skeletal muscles. Muscular dystrophy is progressive degeneration of skeletal muscles, mostly due to genetic disorder. Tetany, rapid spasms or wild contractions in the muscle due to low calcium ions in body fluid. Arthritis, it is the inflammation of joints. Osteoporosis, age-related disorder which is characterized by decreased bone mass and increased chances of fractures. So the decreased level of estrogen is a common cause for osteoporosis. Gout, it is inflammation of joints due to accumulation of uric acid crystals. So these are the disorders of the muscular and skeletal system. And this completes our description of locomotion and movement chapter. So thank you for listening to the entire topic. If you like this video, share it with your friends and subscribe to the channel. Thank you.